This method can help answer key questions in the field of brain disorder, such as how to identify efficient biomarkers that could be used in disease diagnosis.
The main advantage of this technique is that it could eliminate the reference-dependent problem inherent to traditional EEG analysis.
The implications of this technique extend toward our understanding of resting states networks of human brain.
To begin this procedure, import the raw EEG data to the EEG Lab software. Next, load the channel location file into the EEG Lab software to obtain the spatial locations of these electrodes. To remove the reference electrodes, in the option of Select Data in Channel Range of the pop-up dialog box, select only the recording electrodes and do not select the reference electrodes so that the reference electrodes can be removed. To bandpass filter the EEG data between 0.5 and 80 Hz, in the pop-up dialog box, choose 0.5 for the lower edge of the frequency passband Hz and choose 80 for the higher edge of the frequency passband Hz. Then click OK. To remove the power line noise with a notch filter between 49 and 51 Hz, in the pop-up dialog box, choose 49 for the lower edge of the frequency passband Hz and choose 51 for the higher edge of the frequency passband Hz. Then, select the option of Notch Filter the Data instead of Passband and click OK. To remove eye movements, click on Tools, then click Artifact Removal using AAR 1.3 and EOG removal using